So today's topic is monitoring of environmental process with biosensors. So while discussing that topic, we need to understand why we are monitoring the environmental processes or what type of processes are to be monitored and what are the different methods for monitoring the environmental process or environmental status or pollutants status and other things. Then what will be the advantages of the biosensors, or what are biosensors, or what will be the advantages of the biosensors over the conventional methods that is available. So these things needs to be discussed, and not within a single lecture, within one or two sessions we will, one or two or three sessions we will discuss this portion in detail, because this is a topic of relevance nowadays. Okay. So when we talk about process, environmental process or monitoring of environmental process, why we are monitoring the environmental process? Especially, all these things will be related with the environmental pollution. And the environmental pollution that may be defined as the contamination of the physical and biological components of the Earth atmosphere or Earth or atmosphere system to such an extent that the normal environmental processes are adversely affected. So if something, some physical or biological components that is added to the environment, if it adversely affects the normal environmental process, then we can say that that is polluted. The environment is polluted. So that is the definition for environmental pollution. So nowadays we know that the environmental pollution is increasing, and that is actually increasing the environmental pollution issue is becoming or it is increasing in an alarming scale. Okay. So the substances that cause pollution are categorized as actually pollutants. They are commonly classified according to their chemical structure, organic and inorganic compounds, or mode of action like endocrine effect or toxicity, or with the different types of mode of action. And again, based on the source like natural or man-made pollutants, and amount of the pollutants like micro and macro pollutants. So we can classify these pollutants that is polluting the environment based on their chemical structure, mode of action, the source, and the amount of the pollution that is added to the environment. Some of the examples were marked there. A pollutant can be any chemical or geochemical substance, or a biological organism, or a physical substance which has been released to the environment by man and has harmful or unpleasant effect on the or unpleasant or inconvenient effect. So that is a pollutant. Depending on the nature of a pollutant, the pollution is classified as air, water, soil, or land, noise, radioactive, and thermal, etc. There are different types of pollutions, like the pollution or biological pollution. For example, some GMO or some other adversely affecting microorganism that is released to the environment, then that is actually a microbial component that is polluting the environment. And be chemical pollutants that is added, or even noise or radioactive or thermal pollution is also present in the environment. And our focal point is to discuss the microbiological component of the pollutant, how to detect them, and also using microbiological systems as biosensors for detecting the various forms of pollutants in the air, water, and soil, etc. So this will be the focal point when we are discussing the environment pollution and the sensing of the environmental pollution using such a biosensors. So the microbiological, we are looking in the microbiological point of view. What are the microbiological pollutants and how you will use microbiology or related remedies for screening and solving the environmental pollution in different forms. Now, the release of the pollutants, example, heavy metals, pesticides, drugs, and pharmaceuticals to the environment is a worldwide problem, and there is a growing need to combat this and control the pollution. Nowadays, the pollutants, or all the forms of the pollutants like metal, or pesticides, or drugs, or pharmaceutical, radioactive, all the different forms of the pollutants were periodically added or continuously added to the environment, and it is becoming a worldwide problem nowadays. So it is a very serious issue that needs to be addressed worldwide to for a better environment. And the Global Environment Monitoring Plan, that is GEMP, for persistent organic pollutants, which is also known as POPs, has become an important component of the evaluation of the effectiveness of the Stockholm Convention. The Stockholm Convention is actually it's in I think May 17th of 2004. That isn't let's say an international treaty or an agreement between the nations. Stockholm Convention for the control of persistent organic pollutants. So these environment monitoring plan is of these POPs is a major point of which is discussed in the Stockholm Convention for the control of these POPs. And nowadays, these Stockholm Convention for the effectiveness of the or for evaluating the effectiveness of the Stockholm Convention and the remedies that is taken by the different nations for addressing this POP issue is a major area in which we want to discuss the environmental pollution and its biosensing or different types of screening methods. When we are discussing that one, we need to address this one also, because Stockholm Convention in 2004 is actually it's aimed to address the issues of the persistent organic pollutants. And if you want to see how much effective the Stockholm Convention and the decisions or resolutions were, then we need to look into the effectiveness of the different methods that is employed by the different nations for addressing the organic pollutants or persistent organic pollutants. And there are different traditional methods for environment analysis. The classical method or the traditional method that is widely used were chromatography techniques like gas chromatography or ultra high performance liquid chromatography, which is coupled with the MS, that is a mass spectroscopy. And there is MSMS also, that is a mass spectroscopy, again coupled with another mass spectroscopy, MSMS, which means initially you will have a liquid chromatography in which the analyte or the sample that you want to analyze will be fractionated in liquid chromatography, and each fraction will be given to the mass spectroscopy. And the mass spectroscopy will break up these different components or different fractions into small ones, and this mass is evaluated in the mass spectroscopy. And each fragment which is created in the mass spectroscopy is again fractionated in the MSMS, that is the second mass spectroscopy, into which the fractions created in the first mass spectroscopy is given as an input. And in the second mass spectroscopy or MSMS, we will get very accurate mass of each and every fraction which is created from that sample. And based on that fraction or the masses of the different fractions, a unique pattern or fingerprint of the concerned pollutant is generated, based on which the identification of the compound is or the chemical compound is actually done. This is a technique of liquid chromatography which is coupled with the MS. And there are thin layer chromatography also, and other different forms of photometry is also employed for detecting the traditional detecting these pollutants, environmental pollutants, and these are the traditional methods for environmental analysis. And there is when we come to the microbiological pollutants, the gold standard, it is considered as microbiological cultivation. So if you want to detect the presence of a microorganism, certainly the best is to cultivate them. However, the major issue with the cultivation problem is that most of the microorganisms which is present in the environment were unculturable. Only a very small portion were culturable, as you know, because the optimum growth conditions and the optimum media were unknown to the cultivator. So if you know the best condition or best formula of the medium for an unknown organism, then certainly you can grow it. But since the common media like nutrient media or some other specific media that is used in your lab may satisfy at least 5 or 10 organisms which is present in the sample. but the remaining ones will be unsatisfied. Not naturally, they will not be growing in the medium because their specific demands were not met by the medium that is provided, and the growth conditions, including the temperature, pH, and the various growth conditions, were not satisfactory to that organisms. So microbiological cultivation, even though it is considered as a gold standard, but for unknown pollutants or unknown microbiological entities, such cultivation mechanism cannot be used because of the problem of unculturable organisms which is present in the environment. Now there is another method like a DNA-based molecular diagnostic, which is nowadays very popular, and it will give you a better results when you are comparing, when you are considering the case of the unculturable organisms. If you have the specific primers, or even you can use 6S sequencing studies for amplifying the DNA of the uncultured organisms. So it will give you better results when you're comparing it with the microbiological pollutants. And these DNA-based molecular diagnostic methods were also popular nowadays. And with the DNA-based molecular diagnostic methods like PCR, it is possible to identify specific bacterial strains. But this method still requires several hours to obtain the results, and sometimes it fails to discriminate between the related species and the intergenomic heterogeneity. And that means if the DNA, for example, you are going to screen an organism based on 6S RNA sequencing. 6S RNA can be only, or 6S RDNA can only identify up to genus level, not up to the species level or strain level. So a related species, for example, all the bacillus will be amplified simultaneously, bacillus subtilis or anthracis or whatever, everything will be simultaneously amplified by the 6S RNA, and the presence of a particular amplicon or DNA amplified from a sample using a 6S RNA primer doesn't imply that the concerned species is present. Because it gives specificity only up to the genus level. So we can expect various false positive results, or a related organism may be amplified, or a similar organism may be amplified in the sample when you are using such a PCR based methods like 6S RNA sequencing. So this is one of the limitations, and also it takes several hours to obtain the results. A quick identification or rapid identification is not possible. Why you require a rapid identification in both the cases, like microbiological culturing, cultivation, and PCR based amplification, which may require several hours. Sometimes PCR amplification requires a pre-culturing or pre-enrichment of the sample, because the original, for example, you are getting a water sample from an environment and you want to see whether some specific strains of the organism or some specific contaminants were present in the sample or not. So you can go for microbiological culturing, but if you are not satisfying the requirement of the organism, they're unculturable. And in case of PCR, since the sample contains only very small quantities of the cells, maybe 1 or 2 per ml of the sample, so the PCR may not result in sufficient quantity of the DNA extraction procedure, may not give you sufficient quantity of the DNA to be amplified using PCR. So what happens is you will get a negative result. The negative doesn't mean that a low quantity of the or undetected trace quantities of the cells were present, and that is not actually detected based on the normal PCR method if you are using. But there are specific kits available, like Coagen, and they are actually manufacturing kits. Maybe 1 or 2 m the sample may be detected using specific kits, and that is actually for leptospirosis, for example. About 20 years back itself, they state that up to 1 to 2 cells per ml of the lepto can be detected using their Coagen kits. And it is possible, but the specificity of the amplification and sensitivity of the amplification is actually questionable when you are doing that analysis with very low quantity of the cells in the sample. One solution for that one, For example, if you are in our the lab itself, Ambali, for her research work, she is analyzing the presence of about 7 or 8 bacterial strains in the water sample using molecular diagnostic methods. And the main issue is that maybe the water sample contains very trace of very few cells. In that case, directly going for a DNA extraction from the original water sample will give you very low quantity of the template DNA, which may not be sufficient for amplification, which may require a pre-enrichment. So for one day, maybe she will enrich the original sample, then she will try to extract it, extract the DNA and try to identify this, which will take more. For example, the pre-enrichment requires 1 hour, and then DNA extraction and the subsequent PCR and gel electrophoresis, that steps also requires more time. So both the methods, like microbiological methods, have their own built-in screening for microbial entities which is present in the mineral samples. Second, these environment monitoring is required because of the increasing pollutants in the recent years. The presence of the increasing quantity of pollutants in the recent years demands continuous monitoring of the environment to identify potent pollutants which is present or newly added environment, so that effective remedial measures can be implemented. And the requirement of new detection systems is actually a very necessary part. As I mentioned, when we take the example of take the case of these conventional methods or traditional methods, it requires more time. And if a water sample, for example, it is severely contaminated, and your analysis procedure actually, when it requires 1 or 2 days for the identification, by the time the organism might have gone out of your hands. So in that case, what happens is your actually it didn't work out, because by the time you identify the pollutant may have entered its targets. So for effective, you require a rapid method of detection of the environmental pollutant. Even though it is microorganism or chemical pollutant, you need to detect these the presence of the pollutant very rapidly using new detection method, and that's why we require effective rapid detection methods or rapid new detection systems for addressing such pollutants. And there comes the importance of the biosensors. Biosensors have several advantages over the conventional systems, and one is their ability for portability. Usually, if you are depending on an LCMS or they are large permanently installed some laboratory, and you can only take the sample to the lab and do the analysis. Whereas the biosensors can be miniaturized, and it can be taken to any outdoor facilities for easy monitoring. And one of the examples of the biosensors is that you might have come across the glucose strips for glucose detection. That's based on biosensor technology. You don't have to go to the laboratory for detecting, or you don't have to go to the laboratory where sophisticated instruments were present. You can do it in your own hand. You can take it anywhere. So the portability of the device, that is one of the advantages of biosensors. Another one, miniaturization. It is actually miniaturized, and compact structures were created using biosensors. And another one, measurement of the pollutant is minimum samples is possible. So at minimal quantity of the pollutant, the biosensors will help you to identify. And there is nowadays multiplexing provisions were also available for different biosensors. For example, if you want to identify multiple targets, multiple pollutants within the same test, and that is it's like multiplex PCR. these new devices or biosensors can be designed in such a way that multiple target pollutants can be identified using a single system. So multiplexing is also done in case of biosensors. So these are some of the advantages when you are comparing biosensors over control methods. Hello? കേൾക്കാമല്ലോ അല്ലേ? Hello?
Hello, sir. Hello, sir.
Am I audible?
Yes, sir.
Any doubt?
Sir, കേൾക്കാൻ പറ്റുന്നുണ്ട്.
Okay, fine. Doubts ഒന്നുമില്ലല്ലോ പറഞ്ഞതില്? So this is an introduction part where we are going to enter the actual theoretical aspects of the biosensors. We are just comparing the biosensors and their application. Why biosensors were required for environmental monitoring, and some of the limitations of the traditional systems like microbiological cultivation and PCR-based technology is already discussed. So when we come to the biosensors, a biosensor is a chemical sensing device in which a biologically derived recognition entity is coupled to a transducer to allow the quantitative development of some complex biochemical parameters. So it's actually biosensor is a chemical sensing device coupled with some biological entity. So there is a chemical sensing device, also there is a biological entity, and there will be transducer which will actually convert the signal, chemical signal to biological signal, or biological signal to a chemical signal or electrical signal. So based on the nature of the sensor, there are different types of sensors are there, and the sensor can be a biological sensor. So the biological activity can be converted to a physicochemical activity, so that it can be detected by a detector. And that conversion of the biological signal or biological reaction signal, a chemical or physical signal, and like electrical signal, is actually done known as a transducer. So a device like is actually designed with a biological derived entity and the sensing device, and this transducer component which will convert the signal, biological signal, or the chemical, biological means chemical signal. That chemical signal is actually converted to a physicochemical signal like electrical signal, so that a detector can detect signal using this biosensor. So this a biosensor is an analytical device incorporating a deliberate intimate combination of a specific biological element that creates a issue. So there's a biological element which actually recognize the pollutant or whatever the entity that you want to recognize. So biological event will be there, and the physical event that transduces that to an electrical signal. So that is the main component of the biosensors are known as immunosensors, optrodes, chemical canaries, then resonant mirrors, glucometers, biochips, biocomputers. So various types of biosensors were already known. So when we consider that structure, it is having two parts. That is a bioelement and a sensor element. Biosensor includes a bioelement and a transducer or a sensor element. So the pollutant is actually the analyte that you want to recognize from the environment. The bioelement will recognize the presence of the pollutant based on a biological reaction. So once if there is a specific interaction between the bioelement and the analyte, it will give a signal to the transducer element, which will convert the the transducer will actually, it will convert that biochemical signal into an electrical signal, and that can be detected by an electrical device. So this is the base concept. It is having two parts, that is a bioelement and a transducer. Then, as I mentioned, there is bioelement in the biosensor. There is a bioelement and a sensor element. The bioelement may be an antibody, or a nucleic acid, or a tissue, or a microbial cell, or a polysaccharide. Anything can be added as a bioelement, which will have a specific interaction with the analyte or the pollutant that you want to identify. Maybe the pollutant may stimulate an enzyme or enzymatic cascade. Then based on which the sensor element will detect that enzyme reaction. Or an antigen antibody like interaction can be used, or a nucleic acid detection method like hybridizations can be used. Or there are tissue-based methods or microbial cell-based methods, polysaccharide binding methods. So different forms of bioelements can be added as the bioelement so that it will identify or recognize the concerned analyte or the pollutant which is present in the environment. Then the sensor element may be an electric potential, because the biological element, while it recognizes that analyte, it will create a signal, a biological signal or a chemical signal, and that will be identified and by the or that will be converted by the transducer or the sensor element into an electrical potential or electric current or electric conductance, electric impedance, or mass, temperature, viscosity. All these are the physicochemical element, or the outputs that is created from that biological element. So it will be converted by the transducer into some other forms, detectable forms by a device like electrical potential, current, or conductance, impedance, like that. So that type of conversion is done in the biosensors. So here we have an example in which the analyte is shown at the left side, and there is a bioreceptor or the biological element that may be an enzyme or antibody or organism or some other receptors, which will recognize the analyte and interact with the analyte. Then during the interaction, if there is an interaction between the receptor and the analyte, then it will give a signal, a biological signal, biochemical signal out of that bioreceptor. Then the transducer component, which will receive that signal from the biological element, and that may be an electroactive substance or a pH change or a heat or light or mass change. So that different types of outputs will be coming out of that biological element, and that will be detected by the transducer, and it will be converted into an electrical signal like an electrode or a semiconductor or pH electrode or transistor or photon counter. Different types of transducer elements will be there, which will detect that signal which is coming out of that transducer. Then finally, all these things will be converted into a measurable electrical signal that can be read and stored by the devices that you are using. So this is a concept of a biosensors, in which a biological element actually identifies the presence of the analyte in the environment based on a biological reaction. And that biological element, once it identifies or interacts with the analyte, it will give an output, a biological or biochemical output, which will be recognized by the transducer and converted into an electrical signal. And that electrical signal actually is identified, or it is analyzed or stored or processed by the electrical devices that is attached to that sensors. Okay, is it clear? Is it clear?
Yes, sir. Clear.
Okay. So this is a very detailed schematic outline of the biosensor, in which from the left side you can see there is an analyte which is coming, and there is a membrane through which it passes, and then there is a sensing part in which analyte interacts with the biological component, that is the sensing part. Then there is a further the product passes through the after the interaction, the product passes through the inner membrane and reaches the transducer. There is an amplifier for the signal, and finally there is a data processing and digital readouts and recorder. So that is the mechanical or physical components which is finally present. So this is how a complex system of a biosensor is actually designed. And this is classification of the biosensors according to the transducers. There are different types of transducers and different types of biosensors have like biomimetic based on then electrochemical and then fluorescence based, optical, temperature, piezoelectric, then immunosensor. So different types of biological, after the identification of the analyte by the biological component, different types of output may be expected. That may be an bioelectrical or electrochemical signal, or a fluorescence, or an optical difference, or a temperature different, or immunoreaction. Different types of outputs will be received, and that will be converted to the such a physical signals like amperometric, conductiometric, or potentiometric, and resonant mirror, then fiber optic, and one optical part. From the optical part, the most popular one is a surface plasmon resonance, or SPR, and that is a very advanced instrumentation, and it is having a lot of applications. I will show you what is SPR. So there is a surface plasmon resonance, which is actually based on an optical output, or optical signal, which is coming out of the biosensor. So in that case, but all the other case, there is a physical chemical signal. But in this case, for the optical, like SPR, there is an optical output, and that will be converted into a signal, and the instrument is known as a SPR. So we have a lot of different types of outputs are there, and for each and every output, based on each and every output, the transducer components may vary, and we have amperometric, conductiometric, potentiometric, and also different types of optical output based transducers are also available. So commonly used systems, we have optical and then electrochemical. We'll see some examples, and I will show you a video.
Surface plasmon resonance, or SPR, has emerged as a powerful optical detection technique for studying label-free biomolecular interactions in real time within a variety of diverse applications, such as life science, electrochemistry, chemical vapor detection, food and environmental safety, and beyond. Due to its simplicity, nearly every commercially available SPR instrument uses a detection scheme called the Kretschmann configuration. In this setup, a light source passes through a prism, reflects off the backside of the sensor chip surface, and into a detector. At a certain incident angle known as the resonance angle, light is absorbed by the electrons in the metal film of the sensor chip, causing them to resonate. These resonating electrons are also known as surface plasmons, which are sensitive to the surrounding environment. The result is an intensity loss in the reflected beam, which appears as a dark band and can be seen as a dip in the SPR reflection intensity curve. The shape and location of the SPR dip can then be used to convey information about the sensor surface. SPR technology is commonly utilized for the study of molecular binding interactions between free analyte molecules in solution and probe molecules linked to or immobilized onto the sensor surface. As a molecular binding event takes place, the angular position of this dark band shifts, and a shift in the reflectivity curve can also be observed. This is a direct method of detection which avoids the drawbacks of labels. Another powerful aspect of SPR technology is in its ability to observe the time-dependent binding interaction between molecules. By monitoring this change in SPR response over time, researchers can study the kinetics of molecular binding events. In the first example, an analyte with low affinity to the immobilized ligand on the sensor surface is introduced, and therefore negligible binding interaction is observed. However, when an analyte with higher affinity to the immobilized ligand is introduced into the system, then binding events are more readily observed. Initially, there are many binding sites available, resulting in a rapid increase in SPR response that occurs as analyte begins to bind to the ligand. As the sample is continually delivered to the sensor surface, analyte molecules continue to bind, and the number of available binding sites decreases, corresponding to a decrease in binding rate. As the SPR response levels off, the system approaches equilibrium, and the number of molecules binding and binding become equal. When no more analyte is introduced into the system, the molecules will continue to unbind, resulting in a decrease in SPR response. The association constant, Ka, can be extracted from the behavior of the binding response, and likewise, the dissociation constant, Kd, can be extracted from the unbinding response. The ratio of these two rate constants can yield the binding affinity of the system. The information that can be obtained from SPR serves as a simple, precise, and label-free technique for quantitative analysis of molecular binding interactions.
So that is a surface plasmon resonance, which is actually a technology based on the optical detection methods, in which the there is a source of light, and which will be reflected when there is an electrical change that is created by the binding of the analyte to the surface. So on the surface, which is actually maybe a gold-plated surface or something, onto which the receptors will be bound, and if the analyte actually binds to that part, then that binding will result in an electrical potential change on that or the surface plasmons, so which is actually flowing through the surface, and that change will result in the deflection of the light that is actually passed through the prism, and which will result in the change in the electrical output that is created for the graph. So that's how the surface plasmon resonance actually it works. And one advantage of this SPR technology is that you don't have to add any markers for detection. Usually, such detection methods, biological detection methods, may require some binding of the markers or some probes for identification. But here, that is not required, because only target receptor interaction on the surface of that SPR plate that will result in the electrical deflection, and subsequently that optical path variation or the deflection in the optical path will result in the identification of the analyte and the concentration of the analyte. That's it. So usually the other methods may require some probes or some other markers which is actually bound to that system, but here it is not required. So that's one of the advanced techniques that is nowadays used. So the optical transducer based technology, the classic, the best example is the SPR technology. Then there are electrochemical systems, electrochemical device based transducers are also available. So this is the optical biosensor based SPR, a schematic for that, in which there is a laser diode which actually passes the light through the prism, and there is a flow channel in which the analyte will flow through, and on the surface of the plate, which is a gold film on a glass cover slip or something, and on which the receptor or substrate is actually attached, and the analyte will actually interact with the substrate, and the output will be will result in a change in the surface plasmons, and that will deflect the light, and the corresponding output will be received by the photodiode array, and that amplifier will pass that one to the microcontroller and the computer. So the signal will be received and recorded in the computer. So that's how the optical biosensor based surface plasmon resonance works. When we come to the electrochemical biosensor, the same substrate will be actually linked on the surface on a dielectric surface gate. Then the flow channel, through the flow channel, the analyte will come and it will go out, and during that time it will interact with the surface, and which will result in a biochemical change, and that will pass an electrical potential, and because there is a positive negative it's a dielectric substrate, so that that will result in a potential difference or electrical charge, will create electrical difference, and that will be detected through and passed to the biopotentiostat, where that electrical signal is actually detected on the binding of this substrate. So the electrical signal that is created on the binding of the substrate, that will be analyzed and detected by the transducer element. Then we'll discuss a little bit more about the enzyme electrodes. Okay, for the time being, it is because enzyme electrodes may require a little bit more time that we will discuss in the next session. Okay, so the optical biosensor based plasma surface plasma resonance is already covered, and there are various types of electrochemical biosensors, and enzyme electrodes is one among them, and we will see different types of enzyme electrodes and their applications in the next session.